Hello, this is Dr. Pierce. Welcome to learning about renal function for the CPR1 course. Similar to how I organized material in neuroendocrine, I have a series of videos that are pre-recorded ahead of time for lecture to allow you to preview material if you desire, or give you an opportunity to listen again after class if you desire as well. So stay tuned. This is going to be broken up into a series of individual videos to chunk up the material and to more easily digestible elements. Here are your learning objectives for the entire lecture. I'm going to again break these up into a series of more easily digestible videos based on chunks of learning objectives. And here is learning objective 1, which talks about the glomerular filtration barrier.
What you are seeing here is an electron micrograph of the glomerular filtration barrier. The glomerular filtration barrier is made up classically of 3 primary layers, and they are in order going from the blood side to the urine side, if you will: the capillary endothelium, the glomerular basement membrane, and the podocyte epithelium. So if you are a molecule or a droplet of water or whatever it is that's in the bloodstream that's going to get filtered, you would begin your journey over here on the capillary side. So this is representing the capillary lumen. You would pass through the pores of the capillary endothelium, make your way across the glomerular filtration basement membrane, and then squeeze your way in between the slits of these filtration podocyte filaments here. So these represent the podocyte epithelium, and this would be the directionality of your journey. I really like these other electron micrographs because it's giving you a little bit of a 3D view or a bird's eye view of what the glomerulus looks like. These things are classically really hard to visualize in the mind's eye. And so I want to make sure that we point out a couple of areas here. If you begin up here in this upper left-hand side of your screen, this is kind of the big ball or coiled up glomerular blood vessels, if you will. And as you can see, it looks like a big twisted ball of yarn or something that your cat would play with. And if you were to look at this kind of inset square and then zoom in real close, that is what this right here is representing, this additional zoomed in micrograph. And what you'll see is you'll see the blood vessel coiling around here. So that's what this tube-like structure is. So that's the glomerular blood vessel. And so you're outside of the blood vessels looking down upon them. And what you see is you see this what almost looks like an alien structure, but it is a podocyte. So there's your podocyte cell body, and the podocyte has these arms, these branches that extend off of it and wrap and literally hug its way around the outside of that blood vessel. And you can see the intricate, detailed, just complex beauty of this podocyte and all of its little branches. So this number 1 here is indicating a primary branch off of the cell body, and then the number 2 is a secondary branch off the cell body. But what you'll see eventually is that there are many, many, many tiny little twig-like branches that eventually become what is really the tight barrier that's hugging the outside of this blood vessel. And then you look at this box right here, and you zoom in, and that's what you see in this inset picture, and you can see those finger-like interdigitations. And so when we say that they are interdigitated filaments, they really do look like fingers that are interdigitated together. So this is that exterior portion of the blood vessel. Okay, so if you were floating up above, hovering within Bowman's space, looking down upon the blood vessel, this is what you would see. Now, let's pretend that you are Ant-Man, or you have the ability to zip inside this blood vessel tube, and you are living and floating around inside the blood vessel tube, and you're looking up at the ceiling or the inner walls of that blood vessel tube. This is what you would see. So you wouldn't see the podocyte filaments because that's outside the tube. This is the interior of the tube. So if you were to look up, you see all these little holes here, and it looks almost like a honeycomb or a beehive. And those are the endothelial cell pores that you would classically think of as making up the interior of a capillary lumen. So the takeaway from all of this is in order to get filtered from the blood side into the Bowman space, so from inside this tube, to get out of this tube, you would have to be a molecule that is going to be small enough to fit through these pores, get past the basement membrane, and then squeeze out between these interdigitated fingers of the podocyte. And that cutoff is between 20 and 42 angstroms. So molecules less than 20 angstroms are freely filtered. They're definitely small enough to fit through. Molecules bigger than 42 angstroms are definitely not filtered. They're definitely too big to fit through. And then kind of depending on other characteristics, which we will talk about in a minute, if you're in between that size, you may or may not get filtered. Which brings us to learning objective 2.
Now, you may have learned from anatomy and histology that basement membranes have sublayers, and the glomerular basement membrane is no different. It has 3 sublayers that vary based on density, and they are the lamina rara externa, which is located here or on the outermost or the external part of the glomerular filtration barrier, the lamina densa, which is located here, and it is so named because it has this kind of electron dense area that shows up on this electron microscopy. And then the lamina rara interna located here. So this is internal. So this side of this graphic is depicting the blood vessel or the glomerular capillary. And then the glomerular basement membrane is here. And then up here are the podocyte foot processes. So whenever something is going to filter, it's going to filter in this direction, generally upward. I see my arrow was a little wonky there. The lamina rara externa, the lamina part means layer. And then the rara here and here is indicating light. So its electron dense areas are relatively light compared to the very electron dense, and that is what the densa indicates here. So very easy to remember what these words mean. This is important to understand now because there are pathologies that you will learn about in CPR2 next year that go over changes or pathological conditions or problems with one area of the glomerular basement membrane or another. And there are disease manifestations that really manifest only in just one of these layers. And so that really highlights not only the importance of all 3 components of the glomerular filtration barrier, but how each filtration barrier component is going to contribute to keeping things out of the nephron tubules that are supposed to stay out and allowing filtration of things that should be filtered. So again, this is the blood vessel side. This would be the urine side or the urine primary filtrate side, and things would filter in this general direction across the filtration barrier. And this is going to highlight the clinical importance of the glomerular filtration barrier. So I'm not going to require so much that you memorize all of the different components, but I did highlight the things in red here, like this content, and this content that you really do need to know for my level of teaching at this point in renal physiology. The lamina rara interna and the lamina rara externa. So these are those rara, the light layers, They are made up of proteoglycans. So their chemical structure is a little different than the lamina densa, or that dense layer. And that's why it looks a little different on electron micrographs. These look lighter because they are made up of, I guess you could say, lighter or smaller molecules and proteins, essentially. So of the proteoglycans, so these are protein and sugars together. So it is predominantly made of heparan sulfate, and that heparan sulfate is a very specific chemical structure that imparts an electronegative charge to the glomerular basement membrane. That electronegative charge, if something is negatively charged, it's going to repel other things of negative charge. And what is a major thing in the blood that has a negative charge on it, a net negative charge? Well, that is protein. Okay, so proteins have net negative charges, and if net negative charges are repulsed by this net electronegative charge, then it's going to just help contribute to keeping proteins out of the primary filtrate as appropriate, especially those proteins that are in between sizes that might want to get filtered, but they get repelled because of this negative charge. Now, the lamina densa, as opposed to the lamina raras, is composed of collagen and laminin. And there are specific structures within these two chains that are affected clinically, and you're going to learn them next year. So next year, when you are learning about different disease states or pathology states that you will definitely learn, you will definitely go over them, you are going to hearken back to this area of renal physiology and the glomerular filtration barrier, and why you need to know that, for instance, the glomerular basement membrane is made up of 3 sublayers.
So once again, here is the inside of that capillary lumen, and you are a molecule inside the blood vessel looking up at your walls, your ceiling here of the space that you're in, and you see these holes. These are the fenestrated pores of the capillary lumen. Now, interestingly enough, if you were to take an electron micrograph, these are both electron micrographs here, and you look at it in cross section, up here, this part is indicating the interior of the capillary lumen. Here is the capillary endothelium, and then your glomerular basement membrane and your podocyte foot processes. So again, this is that glomerular filtration barrier, similar to what you saw a few slides ago. But what this electron micrograph shows that the others didn't is that there's something kind of fuzzy here that's stuck to the inside of that glomerular endothelium. That is called the glycocalyx. The glycocalyx is essentially a biogel. It's sticky, and it's made up of a bunch of different type of compounds that are long chains of proteins like hyaluronin and heparan sulfate and other types of sticky glycoproteins. And you don't need to know the names of them, but what this graphic over here is indicating is that these pores are the origin of these sticky web-like fuzzy stuff that lives on the inside of that glomerular capillary lumen. So these pores aren't just empty holes. And what this means now is that that glomerular filtration barrier actually has kind of a preliminary thing that you have to get past if you are going to get filtered, and that is that glycocalyx. So a few slides back, we talked about that if you were in that in-between angstrom size, that you may or may not get filtered depending on other characteristics of what kind of molecule you are. So this series of graphs here is showing you what that means and what is the implication of that glycocalyx. Sticky glycocalyx. Well, that glycocalyx contributes to what we call a size and charge dependency of filterability. So that glycocalyx being all those proteins have all these net negative charges on them. And net negative charges tend to repel other net negative charges. This first graph here on the left is showing you the size of the molecule. So this is in nanometers rather than angstroms. But you can see that when you're above 4.2 or 42 angstroms here, you're definitely not getting filtered. This is a clearance ratio that on the y-axis. You can just kind of superimpose filterability right now because you don't quite yet know what clearance is. But you can see that all these lines go down to bottom, essentially zero. They're not getting filtered if they're above 42 angstroms. If they're less than 20 or so, 22, then they're probably getting filtered, especially because they're small enough to squeeze through. But what you'll notice, however, is that if you are a molecule that has a neutral charge, say you're a dextran or something like that, but if you have a neutral charge, you have this nice kind of sloping line here that shows that you do get filtered, and then you may or may not get filtered, and if you're too big, you're definitely not getting filtered. Interestingly enough, if you throw a net positive charge or a cationic charge onto that molecule, you're actually going to help enhance the filterability, even at the same size. So you can see at a molecule that is 3 nanometers in size, its filterability is kind of low. But if you throw a net positive charge on it, its filterability goes up. Interestingly enough, in the opposite direction, if you throw a net anionic charge or a net negative charge onto that molecule, its filterability goes way down, even if it is technically small enough to fit through. And that is because of the presence of that glycocalyx. That glycocalyx being all of these little proteins and forming a sticky web, kind of like a spider web, is going to catch and repel any other substance that has a net anion or a net negative charge on it. And then this graph on the right here is showing you similar kind of function. If you have a bunch of negatively charged molecules and you're trying to filter them, you can see this line down here in the bottom. This line is the same line as this right here. So these are representing negatively charged molecules that are trying to get filtered. If you do something to disrupt the function of that glycocalyx, and in fact that is what happens in a disease called nephrotoxic serum nephritis. If you remove the negative charge from the barrier, disrupt that glycocalyx, disrupt that negative repulsion, then things that should normally get repelled and not filter are suddenly getting filtered. And the most classic example of that are proteins. So proteins that would typically not get filtered because even if they are pretty small, they have that negative charge, they will suddenly get filtered. And what that means is if proteins are suddenly getting filtered when they shouldn't be, they're probably going to show up in the urine and you get a condition called proteinuria. So in summary, what passes the filtration barrier? Well, you have things that are freely filtered at the glomerular filtration barrier. Water for sure, very small solutes, ions, glucose, single amino acids, for instance. And basically, if they are small enough and they probably don't have a net negative charge, they will get filtered. And then similarly, if they do get filtered, the concentration at the immediate location on both sides of that glomerular filtration membrane should be the same because it's filtering readily across. Things that are not freely filtered are going to be larger molecules like proteins. We call these formed elements, but really cells. So a red blood cell or a white blood cell is definitely too big to get through that glomerular filtration barrier. And if it does and it shows up in the urine, then there's a problem. Technically, very, very minuscule amounts of protein should be filtered.